Hello everyone, welcome back to BioBoost. Today we will discuss ribosome as in cell organelle and how it is functioning inside our cell. In detail we will discuss in this video. So what are ribosomes? We see that they are submicroscopic granules found in all living cells. Usually they are attached to ear or maybe scattered all over it, just like how you saw it in that diagram. They were first observed by a scientist named Claude. Unit wise, based on the sedimentation rate, they are divided into two subunits. One is 70S and one is 80S. Sedimentation is a process by which they get separated, and these subunits are where in prokaryotes and mitochondria chloroplast 70S subunits are found, and in 80S subunit structure is found in eukaryotes basically. We will now see the structure of ribosome and how it is present in our cell. In 3D structure, we see that the largest subunit, which is the 50th subunit, at the base is present, and the above one is a 30th subunit, which you saw is present as a lobular structure. In between center, there is a protuberance, and there are lateral crests, which you can see are present on both the side of 50th subunit, the larger one. There is a body, and the space between it present inside it is a tunnel-like structure. The above one, smaller one is 30th subunit, and this larger one is 50th subunit. We will now discuss a model of prokaryotic ribosome in the next topic, where you can see how 30S and in 50S the 70S subunit are divided. So it is known as the Lake model of prokaryotic 70S ribosome. Discussing it, we see that as it is present in two subunits, the 30th subunit is the smaller subunit, which is usually asymmetric, just like how you saw it in the diagram above, and it is usually rod like. It is divided into two lobes by a deep groove, or it is also known as cleft. The smaller head or the base. Now, the smaller head and also the larger base. Now, small outgrowth is arising from the basement segment and it is called as the platform. Weight wise, it is approximately of 1 million daltons, and it is divided further into 60S rRNA in addition to 21 proteins. Now, in 50S, which is the largest subunit, it is more or less spherical. Also in the diagram you see that two lateral clefts were present. So in addition to two lateral clefts, there is one central projection. It is more or less spherical, and the protuberance that you saw, it is all central projection. Anterior is flat. Weight is 1.8 million daltons, and it is further made up of 23 srRNA and 5 srRNA in addition to 34 proteins. These 2 points you need to remember. What is comprising of 30S and 50S subunit? On a mRNA strand, the smaller and larger one subunits are arranged like this and they make a polysome. Now, during protein synthesis, that is translation, the mRNA is threaded throughout the tunnel region. Means mRNA strand is present all along the tunnel region. Now discussing the eukaryotic ribosome, then it is comprises of 60S subunit and 40S subunit. 60S is the larger subunit present above, and the 40S subunit is the smaller one present below. Now 60S subunit is of weight of 2.7 million daltons, and it is of 28 srRNA, 5.8 srRNA and 5 srRNA in addition to 40 proteins. 40S subunit is of 1.3 million daltons, and it is made up of 18 srRNA in addition to 30 proteins. Now together if we compare eukaryotic and prokaryotic ribosomes, then 70S, that is of prokaryotes, it is made up of 70S subunit and comprises 50S and 30S subunit of ribosome. 50S is made up of 23 5 srRNAs in addition to 34 proteins, and 30S subunit is made up of 16 srRNA in addition to 21 proteins. Means 30S S here denotes the ribosome and it is made up of rRNA and proteins, and these are the units. Similarly, in eukaryotes, 80S ribosome is made up of 60S subunit and 40S smaller subunit, and 60S ribosome is made up of 28 srRNA, 5.8 srRNA, and 5 srRNA in addition to 40 proteins. Also, 40 is made up of 18 srRNA in addition to 30 proteins. Now we will see how ribosomes are aligned to ER. So they are basically attached to the ER membrane. We know that on ER ribosomes are present, and that is what makes the endoplasmic reticulum rough in structure. So we see that the smaller and larger subunit is present. Here the larger one, above one, is of 50S and the down one is of 30S 30S subunit. On the smaller subunit, mRNA strand is present in, and above it are present codons of nucleotides, and in 50S subunit, P site is present where incoming polypeptide chain is extended. Here on tRNA, you can see polypeptide chain is made and is extended. So this one is P site where amino acid chain is present. These are the anticodon in complementary to the codons present on mRNA. Now on A site, another tRNA is present. So here another tRNA is present on A site where the first polypeptide is made of 3 codons. So it is the A site. Now the larger subunit, which is the 50S subunit, is the subunit which attaches to the ER, and it has 2 slots. The 2 slots are P site and A site, which is the donor site and acceptor site. P site is the peptidyl site and A site is the aminoacyl site. Now, amino-acyl tRNA complex is attached to A site, and the tRNA which is carrying the polypeptide chain is attached to the P site. Amino-acyl tRNA complex is attached to A site, while tRNA which is carrying the polypeptide chain, it is attached to the P site. So you can see amino-acyl tRNA A site, polypeptide is P site. The smaller subunit is the one which received the mRNA strand. And the cleft, which is 2 subunits, it lies parallel and it remains attached to the ER. Okay. So next we will see the composition of ribosome. So till now we have discussed that ribosome is made up of RNA and proteins. The different types of RNA and the different type of proteins that we have discussed till now remains the same. So the RNA are RNAs or ribosomal RNAs. They have different sedimentation rates, and the proteins that are approximately 55 proteins, they are categorized as the core proteins and the split proteins. Core proteins are the one which directly bind to the ribosomal RNA, so they are basically called as the primary binding proteins. Core proteins are the one which directly bind to the rRNAs. Split proteins are further divided into acidic and basic proteins. They do not bind to rRNAs, but they interact with primary binding proteins. That's why called as secondary binding proteins because they are indirectly binding with the primary proteins. So they are called as secondary binding proteins. Next, we will discuss the origin of ribosomes, also called as biogenesis of ribosomes. In prokaryotes and eukaryotes, different origins are mentioned, where in prokaryotes it is said that ribosomal RNA is coded from specific cistrons of genome, while in eukaryotes rRNA is said to be synthesized inside nucleolus, which is part of nucleus. Now the proteins which are synthesized in cytoplasm, they get assembled in nucleolus, and there they attach to RNA to form RNPs. Assemble in nucleolus, and there they attach to RNA and form RNPs, which is basically ribonuclear protein particles. Now the ribosomes present inside the nucleolus is called as nucleolar RNA, and it is of 45S. Now 45S is further divided into 42S subunits and 35, 32 and 28 subunits, while as the 20S smaller subunit is further divided into 18S, and they together pass through cytoplasm. Functioning wise, the functional role of ribosome is to synthesize proteins and form long polypeptide chains with the help of mRNA and rRNA, as you have seen in the process of translation, which we will discuss later on. Now, ribosomes are also present inside mitochondria. These are called as mitochondrial ribosomes, and they occur freely in mitochondrial matrix, or they are maybe attached to the cisternae membranes, attached to membranes of cristae. Now, in yeast, fungi, protozoans, and higher plants, mitochondrial ribosomes are of type 70S to 80S subunit, while in animal cells, they are of type 50S to 80S subunits. Their role is to synthesize mitochondrial proteins that form respiratory or oxidative enzymes. Now, chloroplast is also said to have its own ribosomes, which are very similar to prokaryotic ribosomes, and they are hence of 70S type. Now, a structure which is termed as polysome, also called as polyribosome, is also made when multiple ribosomes are aligned in line on an mRNA strand. Also, these ribosomes are dissociated once the protein synthesis process is over. Means it is a temporary structure which is formed when protein synthesis is taking place, and once it is over, it gets dissociated. So this was all about ribosomes. In next video, we will discuss about more CSIR concepts. Thank you so much.